We will start Sandoz Symposium with Professor Dr. Dalia Midhat, Lecturer of Clinical Oncology, Ain Shams University. She will ask a very important question about do we still, there is a role for the first generation anti-epidermal growth factor receptor tyrosine kinase inhibitors in treatment of patients with metastatic EGFR mutant lung cancer, and especially in the low economic countries, and especially in the patient treated under the umbrella of Ministry of Health. Dr. Dalia will answer this question. Please, Dr. Dalia.
Thank you very much, Dr. Diab, for the presentation, and thanks for all my professors and colleagues for being here. I will start my talk with this nice article that was published in 2003, that there is a drug that was newly discovered, and it was published in the Boston Globe, saying that there is a drug that works for some. It was stating very quickly the story of a female patient that's young, never smoker, extremely healthy, non-alcoholic, very fit, that was devastated by being diagnosed as a stage 4 non-small cell lung cancer. With knowing the survival at this time being, it was for 9 months for this patient, for this category of patient. She was extremely devastating, writing her death will, and then she was enrolled in a clinical trial for a new drug that was gefetinib. And for some reason, unknown then, it worked well for her. It worked well for her and for a small group of patients that was initially recruited in the study. And at this time, the researchers didn't have an explanation why this drug worked so well with a small young group of patients and didn't work well for the others. The others started to deteriorate and had the natural survival of the 9 months of the stage 4 non-small cell lung cancer. So they started to study why this group started to respond. We can now this year celebrate the 20 years of the introduction of anti-EGFR TKI in the management of non-small cell lung cancer. The story actually started in 1978 by the discovery of the EGFR receptors, and in 2003, it started the FDA approval of gefetinib for unselected non-small cell lung cancer patients. In 2004, that started to have an approval for erlotinib, and in 2005, they started to discover the EGFR mutation, and the T790M mutation was reported. What we call as the era of anti-EGFR TKI actually started in 2013 by the approval of both afatinib and erlotinib for EGFR mutant non-small cell lung cancer, and as we say, started to have the many indications of anti-EGFR TKI in non-small cell lung cancer, and still ongoing. The EGFR mutations, as it is seen in this, we have what we call sensitizing mutation, which are the exon 19 deletion and the exon 21 L858R. Those are the sensitizing mutation. You can see them in green. Green here. Those are the sensitizing mutations. And here we can see what we call in orange, what we call the resistance mutation. So we have mutations in the EGFR that start to have a good response to treatment, and other mutations that leads to resistance to our treatment. The EGFR mutations, they are divided into common and uncommon. The common is the exon 19 deletions and the exon 21 L858R point mutation, and they both represent around 85% of EGFR mutations. We have the other uncommon mutations, which have the most common of the uncommon, as we say, with the exon 20. And we have the T790M mutations that develop in patients with anti-EGFR TKI who received first generation, and it will be further shown. We have 3 generations till now, with the 4th coming later on in the field, but till now we have 3 generations of anti-EGFR TKIs. We have the first constituted in the erlotinib and gefetinib, we have the second afatinib, dacomatinib, and we have the 3rd generation, which is the osimertinib. The 3 of them, the 3 generations are approved in the EGFR mutation, the most common one, as we previously mentioned, the exon 19 and the exon 21 L858R, but we need to highlight that the only one that's approved in the rare mutation, which is the exon 20, and from exon 18 is the afatinib in the second generation. The first generation, they are reversible. The second and third generations, they are irreversible EGFR binding, with a half-life average in all around 48 hours. And the food effect concerning our first generation, it's our topic, there is no change with food effect. With the highest CNS penetration for the osimertinib, the third generation, and the lowest for our first generation. We have seen the treatment, the first and second and third generation have been tested either against chemo or against each other, and all of them showed in the first line setting an improvement in the PFS. We have one of the major studies that recruited around 1,200 patients, which is the IPAS study. Actually, it recruited patients with adenocarcinoma, never or light smoker, with life expectancy of more than 12 weeks, with a good performance status, with a stage 4 disease or stage 3B that are not feasible for local treatment. They receive either gefitinib or carboplatin paclitaxel. And the primary endpoint was the progression-free survival, with a secondary endpoint of objective response rates and overall survival. We can see here in the curves that there is, for the patients who, for the overall, there is an improvement in the PFS, which was statistically significant. But especially in the EGFR mutant patient, we can see that the curves are well separated and we have an improvement in the PFS. While in the EGFR mutation negative, in favor of gefitinib, while in the EGFR mutation negative, actually, they had a detrimental effect, the use of anti-EGFR TKIs. So it's seen here in the curve that this curve, these curves are the patients receiving first generation anti-EGFR TKI gefitinib. For EGFR mutant, they had the best improvement in the PFS, and they do a lot worse with the chemotherapy. While in the patients with no mutation, actually the anti-EGFR TKI had a detrimental effect. They had the worst survival. So, although the overall survival, it was not statistically significant and was comparable in both, but was all in no subgroup analysis for the overall survival. What we need to know, actually, even in the EGFR mutant, we can see here in the curves that even in the beginning, we have patients that are EGFR mutant positive, but actually they will do poorly, even when anti-EGFR TKI. These patients need to be identified, they need to be known because they need further treatment and further management. Now came the study that established the third generation anti-EGFR TKI as standard of care, which is the FLORA study. It was osimertinib versus erlotinib or gefitinib in EGFR mutant advanced non-small cell lung cancer, and it included only EGFR exon 19 or 21 mutant with stable CNS metastasis. The study actually included around 556 patients, and patients were stratified by the EGFR mutation and race, and the treatment was received until progressive disease. Crossover was also allowed. We need to highlight that crossover was allowed with patients who progressed on the first generation and had T790M mutations. The survival curves, we can see that there is an improvement in the PFS for the favor of osimertinib of 18.9 months, where the comparator EGFR TKI, which was 10.2, and we have an updated overall survival that showed indeed improvement in the overall survival in favor for the osimertinib with the comparative EGFR TKI, 38.6 versus 31.8. But we can see at the end of these curves, the patients that reached up to 48 months of survival, they did well in both, either receiving the third generation anti-EGFR TKI or in the first generation anti-EGFR TKI. So we have a subset of patients that still not identified that do well in both subgroups, in both type of treatments. Concerning the PFS, when they were stratified by the EGFR exon 19 deletion and 21, it is seen that the patients with exon 19 deletion, actually, they had a better PFS of 21.4 months on osimertinib, comparable to those who had a 21 L858R mutation, who had a survival of 14.4 months, even on the osimertinib. So this category of patients seems to not do very well, even with the third generation. We can see here in the subgroup analysis in the first plot that In the Asian population, and specifically the Asian population, because they are very specific in the incidence of EGFR mutation in this category that reach up to 62% in their population, we can see that the line actually is crossing. Maybe this subgroup of patients is not very well responding either to the third or the first generation similar way, but we don't have a specific study with this setting. Also, we can see in the EGFR mutation, those with X21 L858R, it seems that they may be doing either with the third generation or the first generation anti-EGFR TKI. It is similar. We can see that they are crossing the line. We have the LUXNANT-7 study that was done concerning the afatinib versus gefitinib in EGFR mutant patient non-metastatic non-small cell lung cancer treatment naive. There is no clear difference in the PFS and OS between the second generation and third generation. We have the Archer trial that actually studied dacomatinib versus gefitinib, which is a second generation versus first generation anti-EGFR TKI. And within the inclusion criteria, it was no CNS metastases were allowed. And the stratification was also done based on the race and the EGFR mutation type. And it showed that there is an improvement in both PFS and OS in favor for the second generation. PFS of 14.7 versus 9.2 months and OS of 34.1 versus 26.8. But what is, which raised the question mark in the study also, that we can see that there was subsequent systemic therapy that was done. Patients that had received a second generation anti-EGFR TKI were allowed to, when they progressed to subsequent therapy of first generation, and we don't have, actually there were 59 patients, and we don't have clear data about what happened to the subset of patients. Did they respond or not? Then for the overall survival for intention to treat population, the overall survival benefit was not seen at the analysis that was done on the February 17, 2017, but they started to separate, and we can see the overall survival benefit that in the analysis that was updated in the May 2019, with an overall survival reaching 34.1 for the dacomatinib versus 27 for gefitinib. Again, when we look at the forest plot, we can see with a stratification done based on the type of the mutation, there might be is favoring for second generation in presence of the EGFR exon 21 mutation in comparison to the exon 19. And concerning the survival benefit for the exon 19 deletion, we can see with the dacomatinib a survival benefit of 36.7 versus 38 in gefitinib, while in the EGFR exon 21 mutation, the survival was 32.5 versus 23.2. From these differences in the survival, we might get the doubt that patients with exon 19 deletion are actually doing good with the first generation anti-EGFR TKI as much as they are doing with the second or closely to them. But again, we don't have a specific study for this category. So what we need to say is that we need to identify our target every time. We need to identify which patient will benefit from which drug exactly and when, because in the presence of many choices and in the presence of the variety of the prices, we need to know which patient will benefit of most from the more expensive drug to be able to give it and provide it for them. Here we can see closely difference in the response rate and median PFS and median OS between different anti-EGFR TKI families. With the third generation, we can see the highest overall response rate of 80% and the median overall survival of 38.6. But you can see with the first generation in the gefitinib group, we can see that there is a subset of patients that can reach up to 34.8 months, even with the first generation. So we need to identify these patients because they may benefit exactly as if they are receiving the more expensive drug. But again, till now, this group of patients has not been identified. So as we see in the NCCN guidelines, it is still the standard of care. The preferred option is the third generation, but it's still category 1 to receive either the first or second generation. There is a meta-analysis that was done on around 8 studies This meta-analysis, we can conclude from it some information to identify which patient benefit more from which type of treatment. We can see that it seems from the chemotherapy groups that the tumors with exon 21 mutation L858R, they might have a better prognosis, even with chemotherapy than those with exon 19. But actually, the exon 19 deletion tumors, they do better with the anti-EGFR TKIs than those with exon 21. And we can see in this forest plot from the meta-analysis that concerning patients with exon 19 deletion, all studies were in favor for anti-EGFR TKI, while in the exon 21 L858R, we can see that there is a tend to be more close to the midline, meaning that the benefit is not as strong. It is there, but it's not as strong as patients with exon 19 deletions. Here we can see, this is my last slide. I hope I didn't take too long. This is my last slide showing the available lung cancer portfolio for drugs that are available at time being in Egypt for lung cancer. And we can see here that in November 2022, it was the first introduction of the first generation anti-EGFR TKI in the Ministry of Health. And so those patients will have the benefit of receiving this type of treatment. And thank you very much for your attention, and I hope it was good.
Thank you very much, Professor Dr. Dalia, for this excellent and to the target presentation. And I have only one question. And if there is no finance, and we will start with the first generation TKIs, what do you prefer, or how can we choose between the different first line, first generation TKIs, gefitinib, erlotinib, afatinib? If there is any patient characteristics, any toxicity issues, any type of mutation, or the three drugs are equal in the first line?
Concerning the afatinib, it's a second generation anti-EGFR TKI. It has a more different toxicity profile than the first generation. Concerning the first generation, we can see the first generation gefitinib was the most tolerable one with the least available side effect adverse event reported even in the studies. But concerning the PFS and OS, there is no difference between them.
Okay, thank you.
Thank you.
Any other question for Dr. Dalia?
Thank you very much.
Thank you. And we can close the session now and go to the next session.